All right, so last module, beginner. Hopefully everyone's feeling good. I know I feel a little sleepy. I'm gonna do some jumping jacks. Get ready here. Okay. Okay, so module 4. So this one's actually going to be half me lecturing and then half Ryan lecturing. So I'm going to talk about differential abundance and some functional prediction, and then Ryan's going to talk about some co-occurrence network. The lab portion is a bit sort of choose your adventure maybe a little bit. Like it used to be, it's probably too much unless you're like amazing. You are all amazing, by the way. But I pick what you want to do first and then move on from there. But we'll talk about the lab more later. Okay, so the learning objectives for this is understand what I mean by when I say differential abundance methods or DA methods, the importance of false discovery rate correction, and appreciate some of the inconsistency across different DA methods, which I'll highlight in a manuscript from us from a few years ago. And then I'll go into about how you can possibly predict some functions from 16S data using a tool I developed called PyCrust. And then also, importantly, appreciate the limitations of functional predictions from 16S data. As always, feel free to interrupt and we'll see how it goes. It says turn up volume. Let's see if I can get rid of that for you. No, no, no. Okay, I guess you're going to have turn up volume on there. Okay, so differential abundance testing, what do I mean? So this is the classic question of, okay, we've looked at alpha diversity and beta diversity at sort of community level, right? But what about, what specific bacteria species, right, is statistically different between my groups of interest, right? And so you may have 2 groups or 3 groups of interest, but obviously we can think about comparing across those groups and then looking for taxa that are different. So here's just some stacked bar charts to sort of highlight. Just trying to make this bigger from my perspective. Sorry. Yeah, this will help. So here's just 2 groups. This is tumor tissue and saliva, and it's asking, okay, what taxa are different between this? You might maybe say this big bar of dark blue here. If we use another type of growing between low and high, then you could also look for possibly taxa that are different between those. And so this is things like, a classic t-test, right? Would be a differential abundance method. But you'll see that there's a lot of specific ways to do that that are more tailored towards genomic data and microbiome data. We can also do this at different levels, right? So we collapsed our feature table to genus level or family level, and we can look for statistically different taxa across those different levels. So often would be family or genus level. Often people don't do it at the species level with 16S data just because the portion of your ASVs that have a species level are quite low. You might get like 25 or 30%. So then you could only actually do the test on those. If you're more interested in something specific below genus level, then you might just go directly to an ASV and say what specific ASVs are different between your groups of interest. So that's great, and we'll talk about a few of those methods. One important aspect of that is false discovery rate. So I remember I talked about facts and opinions. I think this one's probably mostly a fact here. Usually, although it's amazing still, there's tools out there that won't apply a false discovery rate, but almost 99% of the time, you should be really thinking about correcting for false discovery rate. And so if you haven't heard of this before in other scenarios, obviously, if we think about classic statistics of using a p-value cutoff of, say, 0.05, if you recall back to some stats history a long time ago, that means that you're rejecting the null hypothesis 5% of the time, and then you're okay with that as your probability. And so that's great for one test. But then what happens if you do that for all your ASVs? Now you have thousands of tests that you're running. And if you do 1,000 tests, that means instead of a 5% chance for that one event, now all of a sudden you would expect getting, say, 50 significant taxa within that randomization, right? So this is a classic problem where the more tests you do, you lose statistical power and you have to then sort of correct for that. And so that's multiple test correction. And so there's a lot of standard methods for that. Bonferroni is a very conservative, meaning that you lose statistical power, and it's very robust. It's literally just multiplying your p-value by the number of tests you do. Usually, most people would apply something like a Benjamin-Hochberg, which is a bit more elegant, and you keep some of that statistical power, and then you'll often report Q values. I'm not going to go into more detail than that. It's just that keep that in mind. You should be usually applying an FDR cutoff. Sometimes that's built into the DA method that you're using already, but it's good to figure that out. So moving on about types of differential abundant methods, and the classic, like which one should you use? We came across this problem a few years back, and we decided to actually take a survey of a whole bunch of different methods and actually basically try to test them out, and the classic, come up with, which ones work the best. So for this paper, we looked at 16S data sets. We actually looked at 38 different data sets with 10,000 samples in total. And for this one, we're only comparing two groups. Okay, so we're just doing two group tests. Obviously, there's statistical tests where you'd want to compare across multiple groups. And we compared a whole bunch of different methods, 14 of them, some of them with also looking at, say, rarefaction and CLR. And many of these are things you might typically see in statistics. Other like Maslin and Lefsey are very microbiome specific, and some of these are coming from other genomic applications, usually like RNA-seq and things like that. And basically, we just wanted to say, hey, how well do these things overlap? And the ones that overlap the best or something should be pretty good. And we're also hoping for something very consistent. And so what we did is we basically, this is a bit complicated to look at first, we basically showed how many ASVs were significantly found different between those two groups across all these different types of studies. So every row here is a different study, and then the values and then the color of that cell in that table represents how many ASVs were significant predicted by that tool. So if we just look at the top one here, this is a human C. difficile study. And we can see right away now that depending on the method, we get as low as 98 ASVs found significant, or as many as 3,312. That's a big difference across a different statistical method. We see that some methods are a bit more lenient and prefer to find more things, which is fine, and we're not surprised about that. Yes, question.
Just make sure these methods are supposed to be interchangeable. You can be like when you're choosing your analysis, you could choose either of them.
Yeah, so that's a good question, actually. So a statistician would say no. But in practice, within the field of microbiome, you could definitely see many of these different methods applied for the same question, right? The same question of being what taxa are different between my two groups. Would you see people apply t-tests? No, not usually, but we just include it for the heck of it. But many of these other tools, Maslin, Lefsey, ANCOM, ADLX2, CornCob, you would see them across manuscripts. And that means that when we talk about biases, right, what you report as significant will vastly be different than what someone else did, even though they had possibly the same data, but they just applied a different differential abundance method. A statistician would come at this and say, well, they make different assumptions about the underlying data, of course, and it's not surprising that they're different. And we don't expect them to overlap perfectly, but I think we were a bit surprised at how much of a range there was here. And some of them were more conservative, so that's great. And we thought that's, maybe it's just a factor of, okay, depending on what you sort of feel like, that sort of false discovery rate side of things, maybe you would want to choose something more conservative or something a bit more promiscuous. But we found, unfortunately, that even the conservative methods didn't always, all their features didn't just form within the more lenient ones. And so what this consistency score is just literally how many of the features, the percentage of those features that were found by that number of tools. So there's 14 tools we tested, and we see that with some methods like this Lima-Voom or Wilcoxon CLR, most of those, 40% or so, or in Lima-Voom, that's more like 60%, most of those were found only by essentially 2 or 3 methods, right? So that's not very great. And then maybe even more concerning is that for our more conservative tools, we'd hope that maybe, 100% of those would have been picked up by all the other methods. But even for those, it's only about 40%, meaning 60% of those features were still not even picked up by the other more lenient approaches. And then the last part of the study, I'm not going to go through a whole bunch of detail. Obviously, you can read it, but we also looked at their false positive rate. And so this was just by taking different data sets and randomizing the samples into two groups, and then looking for significant things, and anything that was found would be considered a false positive. Most of the methods actually did fairly well, so they're close to 0. This is a log scale. But you do see some of the methods here having a few different false positives, and this Lima Voom sort of almost going off the scale with hundreds of false positives in a single data set. Okay, so that's quite depressing. So then the classic question is, you said to me, what should I do, right? And we say, well, it depends, the classic bioinformatics answer, right? And so it does come down to a little bit about obviously knowing whether the tool tends to what it values, being a bit more sensitive, being conservative. And so it comes down to, really your concern about how exploratory is it? Do I really only want to find things I'm really sure is different across the groups? Or maybe I'm just trying to come up with a list of possible biomarkers that I'm going to then, do something further to filter down. And so that's where it kind of depends for you a little bit. Besides that, there is some takeaways a little bit. We found that, I didn't show it here, but that if you remove a lot of those rare taxa using, say, a prevalence filter, that actually helps quite a bit in removing a lot of that variation and noise. So you may be interested in rare things, which is great, but removing some of that long tail, essentially, of more rare things will actually help both with your power and also with some of this variability. And then the biggest takeaway for us was because one didn't clearly outwin the others, and 2, I'm not much of a statistician enough to basically make a new method at this point. I sort of advocate a lot for the use of multiple methods and the reporting of those methods. So we were talking about biodiversity methods earlier and about how many we should apply. Kind of comes into play here as well. And I think the reality is, I think as scientists, we just have to be a bit more honest about our results. And it's okay that it's not super cut and dry, right? We can report that we may not know perfectly how significant it is. And so what would look like in practice, this is just an example I took from Robin's preliminary data on some oral microbiome and mental health traits. And this is not the only other way to do this, but obviously these are 3 methods that Robin used at the time, Maslin 2, ADLX2, and ANCOM. I think we tend to use Maslin 3, ADLX, and ANCOM. Is that what we've been using now?
Yeah, usually they seem to all be relatively easy to run. The only issue is that ANCOM came out with another version and it's like wildly different between the 2 versions, the results. And I'm not really clear on what the differences are between them. And like other people post issues on stuff and like I'm thinking when to get past.
Yeah. Yeah, so it's yeah, so that's fun. So instead of, yes, it makes it a bit more work, obviously. We're not just reporting, a nice list of taxa individually, but this is just showing one way where essentially now we identify what of the 3 tools were significant for that specific feature. And this is a nice example where there's actually pretty good overlap across all 3 tools. We see this one was only picked up, in depression by one method. So yes, it's significant by that method, but maybe we're not as confident in that taxa. And so for the time being, I guess, until new methods, they do continue to come out, and we sort of continue to try them out. I think this is a good approach for now, but that's my two cents there. Okay, so any questions about differential abundance? Great. All right, then I'll move on to something completely different, functional prediction, or what I call functional prediction. If we think about, what we're doing with data, we have our 16S gene and our data, And this arrow represents, all the stuff that you learned yesterday to get to this table. And then this arrow here represents many of the things you learned already today to get some nice visualizations of that data and interpretation of that data. So we have PICO-A plots, and we have some box plots, some stacked bar charts, things like that. So that's wonderful. And the basic, this workshop has been mostly focused on amplicon data, which is great. The advance is going to go into more metagenomics for those people that are sticking around. But metagenomics, data will give you, this taxonomic table as well, as we'll see on Thursday. And we can do some of the same things on that taxonomic table. There's some advantages there we'll talk about later. In theory, we get better resolution, more strain level things, all that fun stuff. But the other big thing is that metagenomics starts to let you ask about, what functions those microbes are doing. And by functions, I mean what genes they contain, because we're talking about metagenomes, right? We're not talking about metatranscriptomics or metabolomics. So I'm talking about genes and pathways here when I'm saying function. But we'll see that, part of that is generating actually a similar table where we have our samples, and instead of counting taxa, we start counting, gene families and pathways. And those can vary quite a bit for those types of pathways. And then we can do very similar visualizations of, investigating those functions and pathways. You do differential abundance on those genes and pathways. And so the tool I just want to talk about briefly here is called PyCrust. And essentially what it does is it lets you take your 16S data and your table here and make predictions based on what we know about reference genome databases to try to predict what this table would look like if we had done the metagenomics. So this was a tool that I developed during my postdoc in 2013, and then my PhD student Gavin Douglas published PyCrust 2 in 2020, and Rum just made a nice database update, which just came out this year. And so I'll just explain the basics of it, what's different with this little SC version, and then some limitations. Okay, so the whole idea from PyCrust or other methods that try to do this is that we use all that information we have from reference genomes, right? So we've sequenced a lot of bacterial genomes, and we know in theory what genes are within those and what genes those belong to in pathways. And we can imagine if we have now a table where we literally represent each gene family of interest. I've listed KEGG orthologs here, which if you don't know what those are, it doesn't even matter. Just think of it as a gene family. And if we count up basically, how many occurrences of that gene family exist within that genome, we'll put a number here. And where it's absent, it's obviously 0. And so you can think about annotating genomes and then literally reducing that down to just the presence, absence, and the number of genes or pathways within those genomes. And so that's our reference database. And then you can imagine coming along with your table that you made from your 16S data, and you've collapsed them down to ASVs. And you have now samples by ASVs, right? These are representing counts of your taxa within your community. And then, the simplest example is, hey, what happens if this ASV is 100% identical to genome X in my reference database, right? Well, you could then maybe say, well, we think that then now this ASV likely has 4 copies of this, 1 copy of this, 2, et cetera, et cetera, et cetera. Of course, what happens if we don't have a good representative of that in our database? Then it gets a lot harder to sort of predict what would be in that genome, especially at something like 80%, which would be really significant. And so basically what happens is PyCrust attempts to use this information to then predict a table. And so just really sort of at a high level, the major steps here is that what it does is it places your reads, your ASVs that you would create, so your representative sequences, into a pre-existing multiple sequence alignment and phylogenetic tree that we've made ahead of time and is included with the package. This process is very similar to what you actually did where you placed your reads into the tree to get phylogenetic tree today to do alpha and beta diversity measurements. And then what happens is within our phylogenetic tree now we have our ASVs decorating this reference tree. We then do what's called hidden state prediction, which just means that now we use that big table of genome by functions as our reference. And for every single function, we decorate this tree. And so these red numbers represent those numbers within that table for the tips of the tree where these are references, right? And then we add in our ASVs as question marks and say, okay, what's the most likely then gene copy for that trait within the tree? Okay. And so if we looked at this example, using various approaches to do this hidden state prediction, just looking at it, you can make a guess that, oh, well, these are all three. The most likely candidate here for this gene copy number would be 3, right? This one is likely to be 2 because these are both 2. And this one over here, well, that's somewhere between 0 and 1. And then that happens, for every gene family. And there's lots of different ways to do that. We actually leverage an R package called Castor, which employs different methods, but we use maximum parsimony mostly because it's fast. And the more elegant or intelligent approaches don't actually seem to get you that much better accuracy. So that's wonderful. So that's sort of how it makes predictions for every gene family, for every ASV. And then after that, you're basically just doing a lot of sort of matrix manipulation. So here would be your ASV table with our counts. We talked about 16S copy number, and I think it was mentioned that PyCrust can correct for 16S copy number. So the default is to do that process. So using predictions from PyCrust, we get the 16S copy number for every ASV. And what does that look like? I just put a big division symbol here, which is not technically correct, but it gets the point across that we're literally dividing our ASVs by that copy number to now have like a corrected ASV table based on the 16S copy number. Okay, so that's still an ASV table, just in theory corrected by 16S copy number. Then that table, this is just the same table duplicated in the bottom left. Then we actually now have the PyCrust ASV predictions. So that's what I just showed you with that hidden state prediction. And that's done for every single ASV, which is considered a row here for every function. And then literally all it's doing is just taking those two pieces of information and multiplying and adding them together to now give us our functions by samples, right? So this 13 of this particular functional gene family, K0001 in sample 1, is just simply derived from all the ASVs, their predictions for that KO family by the number of that ASV within that sample. And so it's 2 times 4 plus 1 plus 1 plus 2 times 2 equals 13. So now that's great. So now we have a table, and we could do, in theory, things with that and interrogate it like we would say metagenomic functional table. That clear pretty much? Not too bad. Okay, great. So what's new with PyCrust 2 -SC? If you're curious what SC stands for, it stands for sugar-coated. Why does it stand for sugar-coated? Because Robin and I spent a lot of time trying to come up with nifty names, and they all failed. And we actually, I don't know if I should tell the story. Yeah, let's do it. It's post lunch, right? Any Americans in here? That's all right. You're cool. I thought it'd be really nifty to call it. What's that? Oh yeah, that's a little bit. I don't care. So I thought it'd be nifty to call it PyCrust 2-MPGA, make PyCrust great again. And we actually had that in the preprint. It was worked it into the abstract, had that great. The timing wasn't great. It was right when it was like post-election, but he hadn't. Yeah.
Like, oh, that's kind of crappy.
Yeah, it's kind of crappy, but we didn't realize how bad it was like gonna get. Like it was like it became not funny actually. So then we were like, and then with the editor it was kind of like, this isn't really funny. I was like, yeah, I agree, it's not funny.
happened as well. We were like, yeah, everything's going so downhill that like, we
Yeah.
don't want to make light
Yeah.
of this.
Why don't we want to make light of it anymore? So, yeah, and then we threw around other fun names, and anyway, we came up with empanada.
Yeah, that was it. Yeah, some people do real backflips to get up with some really creative names.
Yeah. We didn't do that. So we just came up with high crust, sugar coated SC. Bam, done. Okay, wonderful. Doesn't mean anything. But now you know, you have an in on why it's there. Okay, so that's great. So the biggest difference is that the underlying algorithm doesn't change at all. So that's why it's still called just PyCrust2. It's not like PyCrust3 because the underlying mechanisms don't change any. The biggest difference is the reference genome database was updated. And then the one technical sort of other change is that we used to do the placement of those ASVs into one tree. But because we're using the GTDB, the Genome Taxonomy Database, for its genomes, they have two trees. And so archaea sequences are put into an archaea tree, and bacteria sequences are put into a bacteria tree. And then it's just combined afterwards. So you don't sort of see that per se, but it is an important mention. And then so basically Robin, obtained genomes from GTDB. Before this, we sort of manually got them from IMG, and it was IMG was through the JGI. Man, I'm throwing acronyms, just weighing it off, weighing them out here. IMG is the Integrated Microbial Genomes Database, I guess. And JGI is the Joint Genome Institute in the US. But it was a very manual process, and basically wanted to have a better way to be able to update the reference database. If you want to hear more about this, you can just look at our little bioinformatics application note, but really this is just showing the number of taxa that were added at each step and sort of the filtering process. And then you can just see here between the old PyCrust2, and then you're seeing the increase in the number of genomes or the different taxonomic ranks for bacteria and archaea between the sugar-coated and non. And then also, this is just showing the top right, the different types of functional predictions you can make. So you can make KEGG, which I just showed, like KOs, but EC numbers are widely used, and we can map those to things like metacyclic pathways. We will talk about this more in advanced. And then there's other functions included. We did decide to drop a few from the old IMG, but overall, I think these are a better selection. And again, I'm not going to go into this in great detail, but in the past, if you want to read the PyCrust2 paper, we used real metagenomics. We used real samples with metagenomics and 16S data to determine how well they correlated if you use metagenomics versus 16S. And this, we didn't want to do that all over again. We just use simulated samples. So this is showing the average sample on the left, and basically Robin randomly took genomes that look like these types of real samples, and then asked about how well we predicted the genome content for those types of samples. Spearman correlations are shown here between the old version and the new version, and Bray-Curtis dissimilarity is showing the difference using that metric. And the last thing I wanted to mention is weighted NSTI. NSTI stands for the nearest sequence taxon index. And the best way to think about this is essentially it's a measure for your particular sample, how well it's represented in the reference genome database, right? So you can imagine if you have a sample, especially say like human gut, we have lots of good reference genomes. And so the phylogenetic distance to that reference genome would be small. And we basically add all those up and weight them by the ASVs. And so a smaller weighted NSTI value means it's better represented in that sample. If you had something really diverse, like you go to, I don't know, Antarctica and you collect a soil sample or something, that NSTI would be larger because we don't have as many good reference genomes. Okay, so that's great. Okay, so PyCrust. This is how I use it. Yes.
That is one that is we could not classify, or is because the abundance in the sample was low.
For the NSTI?
Always the ones in the graph is some species that you could not classify.
Oh, yeah. Sorry, yep. No, over here. Yeah, that's just a.
Low abundance.
Yeah, it's a common technique that you can sometimes use for stacked bar charts because you'll often get back a big list of taxa. Say you have like all your different genera, and it's quite common for people to just rank the top 15 or so and then group everything else into other, just because visually you can't see like 100 colors on a stacked bar chart. Yeah, yeah. Okay, so pie crust, yeah, so it's an acquired taste. So some people love the tool, yeah, and some people love the pastry. And so it's been widely used. Like I think it's been cited about 15,000 times. So it is a widely used, widely known tool because you can imagine it's quite nice. You have 16S data and then you can predict functions. But I'm not going to ignore the fact that it's also, got a nice following of people that do not like the tool. Okay, so I'm asked about it a lot. I've gotten a few discussions on Twitter or Blue Sky, very nicely about this. But because, there's definitely people that don't like it in varying degrees. And it comes down to mostly the fact that, classic example is E. coli. Okay, so classic example is E. coli, and you think about, hey, you can have an E. coli that is very benign, and is not pathogenic, and then you have another E. coli with 100% similar 16S gene, and we know that it's very pathogenic and it has very different gene content. So people use that as example or others like that, which is absolutely true. And they basically say, well, there's no way you can predict function from 16S. And if you can, it's not really meaningful. And so this is a long discussion that I probably won't get into entirely here, but it is true. It's a prediction method, just like many other things. And so I think you just have to keep that primarily in your head, that it's a prediction method, and you have to think about the samples you're applying it to. So, human samples where we have good reference genomes, definitely, I think it's more acceptable. You start getting into soil samples and there's a bit more of a pushback. And usually, the reason I feel bad about it is, people use it and they write papers and then they get a really crabby reviewer and they're just like tearing it up. And I feel bad for those people. So usually I just try to, get the point across that yes, it's useful and has its place, but there is definitely limitations. And so just to hammer those home here, one, it is a prediction of gene abundances, first of all. So functions already gets a little messy because people jump the gun with, what that means. These, just like metagenomics, we're talking about genes, right? So we don't know if it's transcribed. We don't know if it's that pathway is making the metabolite that we think it's doing. That's a problem outside of pie crust. We know that the accuracy could be low for individual ASVs, especially those that aren't close to a reference genome. That sort of gets averaged out a little bit across the community, is why we do pretty good. And then also when we talk about the accuracy of the method, especially with in the previous papers, we are defining the things that we're predicting as those enzymes and pathways that have already been characterized by the microbial community. And so even though when we sequence a new genome, we know that like 25% of the gene content can vary, with a new strain, the reality is that most of those genes are like not characterizable. They're functionally, we can't annotate them very well. So a large portion of those have unknown function. So because of that, it means that pie crust isn't really talking about, I guess, that cloud of genes that we don't know their function anyway. And so that actually helps a little bit with why we see the accuracy pretty high. So that's limitations, and I think the biggest thing I always tell people is hypothesis generating tool, right? So you do your 16S study, maybe instead of just, talking about the types of taxon, what are different, it's an extra step to say, I wonder if there's something functionally that's jumping out from these types of bacteria that I'm not seeing just by their names, right? And that's really why we developed the tool. So, from a practical standpoint, I wouldn't write a paper that's like 100% based on pie crust results. It'll probably get shot down by somebody, but it often works as a nice, like, hey, this is one of the last figures in my paper. And so these functions could be of interest. And obviously with this, or even as you'll see later on with metagenomics, all of it takes a lot more validation, right? I mean, it's an idea of, hey, maybe I can chase after this and, go down this rabbit hole. So that's my spiel on pie crust. Happy to talk about that more sometime. If there's no questions, I'll get Ryan to come up while I take more questions. Or if there's no questions. Yeah.
Output? Yes, from C, but does it tell you how, like, the percentage of your features that were identified with pie crust? Like, do you know how much of your data set it got info from?
The functional features?
Yeah, like, were all of the ASVs included, or is it only like 20% of the ASVs were?
So the sorry, what?
I said, you want me to take that?
No, I think it's fine. No, I didn't know. Yeah, so all of the ASVs are included now in the prediction because they're all placed into the tree.
But like, what if it's a novel microbe that isn't placed?
But usually, like, almost all of them placed. It tells you when you're running it if any of them complete.
Okay.
You can set a different threshold of how close you want it to be. And you can also look in output and see each ASV how close it was.
But yeah, coming back to your question a little bit, so the ASV, unless it's like really like whacked, like it'll get
Yeah.
placed in the tree somewhere.
It doesn't be 100%.
No,
Okay,
definitely not.
right, yeah.
Yeah, so it's placed in the tree and it's not based on taxonomy, so it doesn't have to have a name or anything, right? It's all phylogeny based, so it doesn't have to have, a genus name or anything. It just has to get placed in the tree.
Yeah.
Obviously, if it's a really new branch, then it's going to have a high nesty value, and obviously the prediction for that ASV will be poorer, but it will be there.
Okay.
And then yeah, I guess I didn't talk about this too much, but like you can stratify for the functions that are predicted. You can look at the output and see what ASVs are contributing to that function.
Oh, cool.
So if there's a function like, oh, this is really interesting, you can at least then connect it back to the ASVs and then sort of do that sort of interpretation too. Yeah. No comments from Steve Helm over here? Which one are you, Steven? Are you this one top one? Or are you the bottom one? It's okay. You won't hurt my feelings. A little bit of both.
Philosophical than practical.
It's okay.
But there is the main challenge to the inference problem that microbial diversity in natural environments at the genome level is very high, and the ASV as a marker for differentiating genotypes is not very good.
Yes.
Like if you look in coli, for example.
The next
That was a good example.
You can have the same ASV that has a genome that's 35%, 40% different at the genome level. And so that's a complicated factor. And then if you look at the population studies, like the Vibrio study, the isolates of Vibrio, where again, it's the same idea, exact same ribosome RNA gene, genotypic variation across 1,000 isolates of 40%. So the genotypic content of naturally occurring population, essentially there's no, and so inferring function based on the ribosome RNA gene in isolation is, I think, challenging.
It's challenging. Yes. Yeah. Do you think it has its time and place? I know I keep pushing you on.
I think anything that builds hypotheses that can be tested is a useful tool.
Okay, perfect. Then we agree with that. Yeah.
A lot of my clients are engineers, and to them, bacterial names mean absolutely nothing. So having something that puts it into like a chemical name, like this probably does something with sulfate, then they're like, I care about this then.
Right.
So that kind of setting, but then you need to be careful with them talking about the limitations.
Yeah. I don't have the slide here, but that's usually how I start the whole idea, right? We label things with taxonomic names because one, we like to track these things, but two, we already sort of infer what that means, right? Like at a high level, not at maybe the gene level. At a high level, we start to go, oh, that's, coli, or, it's Akkermansia, or it's bacillus, or all these names infer some functions already in our head. This just really quantifies it and maybe pushes it to some people a bit too far, but yeah. But but obviously widely used and I think in the right time and place, it's
I
useful.
mean, the desire to do trait-based mapping has existed since we started sequencing ribosome RNA,
Sure.
right? That,
Yeah.
You know, and there are some very strong indicators, for instance, methanogens. Pretty easy to differentiate from a ribosomal RNA sequence, that's a methanogen. Sulfate reducers, pretty easy to say there's probably sulfate reducers. It just gets murkier the deeper you dive into the metabolic network.
Yeah.
Because there's so much movement of genes between paths.
I was gonna use your Zoom.
Perfect. Yeah. Did you send it to me on Slack by any chance? Oh, it's in the thing. Of course, the same thing
It's
that
in the thing.
it's in the thing.
I'm not yours.
It's all good.
Yeah, I think so.
Do you think we did it?
You want to do it here or download it maybe?
Well, yeah, whatever you just did
I would-
seemed like it worked just
Yeah.
great.
And we had it come up. That they were like, oh, what about, why haven't you compared to the other one? But also, none of the implementation.
Yeah, we were just downloading the new versions, but it was.
After that failed.
It does it based on the needs.
Yeah,
that's not
It's very.
Tacked right at the end of yours.
That's not the right one. Did I
It
just
should
tell-
be just scroll all the
Yeah,
way.
just screw, yeah.
Oh,
all the way at the bottom.
I'm sorry.
So,
Yeah, that would have, okay, I screwed up then.
That's all good. There we
perfect.
Thanks. Sorry, I didn't.
go. Okay.
Make sure that I can click. Perfect.
Okay, well, click out more muttering.
Yeah, well, I know, I like the little muttering. It shows that it's a real thing. We're real people. All right, last presenter of the workshop. I have a loud voice, so hopefully I engage you. We have 11 minutes to learn about co-occurrence network. The math is a little complicated, so we might have to breeze over a little bit of that. My name is Ryan. If you didn't already know that, I'm a senior graduate student in Stephen's lab, bioinformatics. I do a lot of different types of things. I do some co-occurrence networks, and if we have a chance at the end of this to talk, I actually have one of our publications where we did some co-occurrence and we paired it with metagenomics and transcriptomics. And so I also, you can read it on your own too if we don't have time to get through it. So, but first, the learning objectives for this slide, if we have time to get through all of it, I'll try to keep us to the time just because I don't want to overload and I want us to have time to actually work through the workshop part or the lab part. I'm just going to talk to you a little bit about the anatomy of what a network is, and more generally what a graph is. I want you to kind of understand what the underlying advantages of doing a network analysis versus some of the things that we already did, and other type of correlation analyses. There are certain caveats to doing, as there are caveats to everything. There's caveats to doing network analysis, but it is a very, very powerful analysis. Then I will talk a little bit about how those particular analyses do apply to the type of work that we do. And then I'll show you, if we have time again, to a little example of how you might deploy something like that within a study. So first, a little history lesson. So in the 18th century, there's a city called Königsberg, and the people of Königsberg liked to go on walks through their beautiful city. And their city had multiple land masses, and it had 7 bridges. So this is the 7 bridges of Königsberg. There was a famous mathematician at the time named Leonhard Euler, and the overarching discussion in the community was, is there a way for me to go on a walk and walk across each of the 7 bridges without walking over any of them once, or more than once? So once and only once. Is there a path through the city that we can do that? And so, although Leonhard was not really interested, he actually was quoted suggesting that it wasn't really a math problem. He was requested to look into it, and he did. And so, by doing that, he started to lay the foundation of graph theory and, by extension, network theory. So he used a method called abstraction, where you break the components of some complex system into the most simple components, and thereby created a graph, which is what we see, the difference between the land masses on the left and the bridges converted into an abstracted graph on the right. And that's what essentially a network is. It's a type of graph where the points are vertexes. They're also called nodes. Most commonly in our field, they're called nodes. And then the edges are the connections of those nodes to each other. And so you can see that some nodes have multiple connections to other nodes, some have singular. So the end result of this, to just give it away, was that there is no solution. Euler was able to determine that if a node has an odd number of edges connected to it, if all of the nodes in a network have, excuse me, let me reverse that. If all of the nodes in a network have even number of connections, then there is a path from one node all the way through to the other nodes to that node again, and once and only once. If there are two that are odd and the rest are even, then excuse me, the first one's called a circuit, the second one's called a path, which is from one node to the other. And then lastly, if there are odd number edges, more than two, then there is no solution. So what he determined, and the proof of this particular theory was actually proven later in mathematics. He didn't actually do that. It was many years later. But essentially his definition was that it was very simple to determine if that path was able to be done just by counting up the number of edges on a node, and then boom. Very, very, very easy to do. Now, as a bonus, if you were to ask someone maybe if there was a way to visit each node once and only once, so that's the completely opposite of the network of edges, that's called the traveling salesman, and that actually is called a Hamiltonian path, and that is actually incredibly hard to do. So that's considered an NP-complete problem, which means that there is no actually easy solution to it. We don't have to go deeply into that, but if you're interested at all in talking about that, it's actually pretty fascinating. Okay, so some core concepts and definitions for networks. I told you about the nodes. Those nodes can be multiple different things with respect to the data that we look at. Networks are used for all kinds of different analysis. You can talk about them with respect to neighborhoods like we did in Königsberg. They can be samples that you're analyzing. They can be individuals within a cohort. They can be individual genes. And by extension, they can be ASVs, which is what we were looking at today. The edges that connect those different types of data can be the bridges of Königsberg. They can be the treatment, the interaction, expression information, or in the case of what we're looking at in the lab is correlation. They can be directed and undirected. So I put them onto the actual graph there. You can see the arrows pointing. So in the first graph that I showed, there was no arrows. That just meant that they were bidirectional. But in fact, you can actually give directionality to those to overlay more information onto the network. You can also weight the edges based on some type of information. In our case of what we're going to be doing today, we're going to be looking at correlation, and correlation can be both binary, just correlated or not correlated, but more importantly and much more useful is to have some type of weight that's associated with that. So you can have stronger correlation, you can have negative correlation or positive correlation, and then you can have also weak correlations. And so then there's, along with being able to actually visualize these types of networks and start to kind of look and explore your data, you can also calculate a number of different really interesting metrics for those networks that you're using. You can do degree, which is the number of connections per node. That was what Euler was using, which was the degree of a node. There's centrality, which talks about whether or not certain nodes are more influential in the network, or there is betweenness, which helps to bridge between multiple nodes across a community. There's clustering, literally just looking at how close they cluster to each other within the network. And I'm going to show you a couple examples of biological systems where you can see clusters and see a number of these different metrics visually. And then there's also path length, but there's a bunch of other types of metrics. These are just some of the more common examples. So therein lies the question of, okay, so it's a correlation network that we're looking at. Why do we need it? I mean, I already know how to do Spearman. I know how to do Pearson. Is there some advantage to doing this wildly more complicated math on my data? And the answer is yes, depending on what you're trying to do. It can be very, very helpful. So pairwise correlations, they measure direct relationships, pairwise, so one-to-one. They can't really distinguish the indirect associations. And anybody who knows anything about microbial communities, there's lots of really complex interactions within these communities. There's emergent properties, emergent metabolisms. There's all types of syntropies and things that are happening. And so these types of pairwise comparisons are not particularly good for trying to glean those types of interactions. It's very challenging. They're also very sensitive to compositionality, which is something we've talked about a lot during this workshop. Amplicon data is compositional, and so these methods are not going to be particularly good. We did show you some methods for transforming your data to help with that, and thereby you can apply some of these pairwise correlations. But if there's other methods out there, maybe we should try those as well. And then they also don't give you very much structure with respect to actually visualizing and then calculating some of those metrics that I talked about that a network can actually allow you to calculate. So the opposite of what I just said, the network analysis can help a lot with these types of things. They can contextualize the relationships at very high levels. At a systems level, they can give you insights into indicator taxa that are particularly important for certain aspects of the communities that you're comparing. They can show you modularity. They can show you clusters, and you can calculate those network statistics. This is a picture. This figure right here came from actually the Speakeasy GitHub. I won't go into detail of what it's showing, but it's human gut microbiome. And what it is showing is it's showing modularity. You can see groups that are collecting based on how similar they are to each other and how they correlate with respect to, and primarily to the abundance of the ASVs across different types of samples. So most of the networks, actually, I think today all the networks I'm going to be showing are not, the nodes are not samples, they're actually ASVs. And so we take all of the data that you created from your ASV table, and instead of showing the samples and showing how similar they are, like what we did with Bray Curtis, we actually show ASVs and how they are associated with each other across different samples. Yeah.
You know, are not clustered, but they are linked together.
Yeah.
Based on how similar they are, but nothing functional.
There's no functional information. You can associate that. There are networks that do that. Yes. In this particular case, it's using ASV data and abundance information across different types of samples.
But I can say that they are the same family or not related.
Well, you can apply taxonomy to an ASV. So yes, you can overlay taxonomic information onto a network. And actually during is that what you're asking?
Yeah.
Yes. So during the workshop or during the lab, at the end of it, you're going to actually take a very simple network and you're going to be able to overlay certain types of information onto that network that is not necessarily part of the calculation of the network, but can give you attributes. So taxonomy, different types of treatments and things like that. You could just color them in it. It's actually very, very helpful. That's what you're seeing here, is that the different colors that you see here are different taxa. I'll skip over the basics of Speakeasy. We're going to be looking at this during the laboratory. But Speakeasy is the actual algorithm that we're going to be using today. There's a number of different algorithms out there. I like Speakeasy because it's particularly good for very sparse networks, or sparse matrices, which is what we have. So I'll just skip over that, but just so you know, this is the one that we're using today, and the actual DOI for the publication is there, and you can find their GitHub through that. It's very, very nice. All right, so applications in microbiome data, we're trying to show these interactions. As I said, we're looking for competition and symbiosis. We're trying to identify microbial consortia and indicator species for whatever particular treatment that we have. One of the cautions that I'd like to really underline, and Morgan has underlined this as well with other things that we do, is that these are not facts. These are all hypothesis generation. So correlation is not causation. This is going to give you ideas about what a community are doing. It's going to help drill even deeper into those interactions, but you have to come up with studies. You have to come up with tests that actually test what you're seeing. So it's very important that this is just a hypothesis generating, although it can be extremely, extremely helpful. And so this network that I'm showing here is actually from a paper that I'm a little bit over, so I'm going to just say it very briefly. Keep going? All right, all right. Well, then I'll just jump right into this. Okay, so networks in action. Networks in action. The colors that you see in all of these figures are just essentially different treatments of an anaerobic digestion system. So what we did is we got a time series data set from a wastewater treatment plant. They had many hundreds of samples, and what they did is they had 4 different treatments for their anaerobic system, and they were looking to maximize methane. So right underneath here, I'll actually point to it right here. These are the configurations, and you can find I have the citation right here, but these are the different configurations of the system. So the question was, how do we maximize renewable natural gas in our system, and can we use microbes to help us do that? So this tree right here shows you the similarity of the samples with respect to the microbiology, that is the ASV network, as compared to the physiochemical measurements. And so what you can see is that they're not one-to-one, but they're not meant to be. What you do see, though, is that the different colors group with each other across. So what that's saying is that the physiochemical measurements that you measure in the system do, in fact, correlate or pair with shifts in the microbial community, which is pretty cool. And you can also see that in the plot right beneath that. This plot is very similar to what you created with your beta diversity. So this is a multi-dimensional scaling plot that has been also the other information that's been added is those physiochemical measurements. So we're not just looking at microbial community shifts in the microbial community, but we're also looking at those shifts in the physical chemical measurements. And you see these little groups of the different configurations of the anaerobic digestion system. So this plot here shows you the different types of outputs from that system. And as you go across time from left to right into the different configurations, you see that things go up and things go down. But it's a what we found was that the system is fairly consistent and fairly robust with respect to the methane production, based on the measurements that we were taking. It seemed like not a lot really changed. You can see that there's a little bit of a spike right here at the end with the SIR, but in reality, this is the percent methane of the total. And what we also saw was that we saw an increase in a significant decrease in the biogas. So total gas was not necessarily increased. So none of this has anything to do with networks, right? But that's where this comes in. So then we wanted to ask the question, how can we use network analysis to maybe get at what is the underlying, we already saw that the microbial community correlates somehow with sorry, I tend to walk out in front when I start rambling. So we already saw in those last figures that the physiochemical measurements in the microbial community are correlated with each other, grouping with each other. So how can we look deeper into the microbial community and try to find what are the microbes that might be indicators for methane production? So this figure is very, very small, and I do apologize for that, but I'll point you to the things to look at here. This is the total community made from the ASVs, where you can see different colors are the different configurations. This on this side is actually the size of these points. It's the exact same network, but the size of the points is the transcriptional activity of those ASVs paired from paired MAGs, so paired genomic information. And the important bit is C and D, where we actually took single nodes that represented hydrenotropic methanogens, and we took the linkages, the primary linkages, so one away from them, and then one more, so primary and secondary, and we looked to see if there were any subsets or any consortia that were associated with those methanogens. And what the short answer is that we did find certain syntropies, possible syntropies. Again, this is a hypothesis. We can't prove it, But we did find a set list of different organisms, different fermenters, different acetogens and things like that that were associated within the network with these methanogens, which gave us the ability to then start to hypothesize about what other tests we might do. Now, we still have a very large amount of data associated with this, including metagenome and metatranscriptome. We just haven't had time to work through it. But this was a really wonderful way to then, the next step for this would be dig into those metagenomes and those metatranscriptomes and try to see if we can more better support these different consortia that are based on these methanogen ASVs. So that was kind of the end of that story. And I think I've talked plenty. So I will say thank you. And if you have any questions, let me know. I'm happy to talk at length about anything and everything. But I think we need a little break and then we can get into the actual exercise.